Sachs, the volunteer moderator for our APBD patient chats. We've met monthly since January of 2014 to help each other by sharing ideas and questions and discoveries, details of having APBD. Let me set the stage for us today. In late 2025, we began learning about biosamples. The Foundation's research manager, Dr. Lindsay Gill, explained various kinds of biosamples to us and how all of our community members would be asked to provide biosamples. Well, today, Lindsay is back with us to again describe biosamples, give us examples of the biosamples that are most important in our community right now, and why it's important that you, and I'm talking about patients as well as certain family members, participate in our new biobank. So I want to thank you all very much for showing up today, morning, evening, wherever you are around the world. And I do want to welcome Lindsay to take the mic. Thank you, Lindsay, for being with us.
Thanks, Harriet. I appreciate the introduction, and I'm really happy to be back again to speak to everyone in the patient chat again and give you an update and some more information on our biobank. So I'm going to share my screen. And looks like that is there. Okay. So as Harriet said, what I'm going to be speaking about today is the importance of participation in the GSD4 biobank. And what that means for you as a participant and what it means for our community with the APBD Research Foundation. So first I want to start off with just some basic background on what is the GSD4 biobank. So a biobank in general is a secure facility that collects and stores biosamples. And as we spoke the last time I was speaking with you, biosamples are small samples collected from your body, like blood, urine, saliva, or nasal swabs, a lot of the things that are collected for routine tests at the doctor's office or at an urgent care. And for our biobank, these samples are also linked to health information and surveys that are collected at the time of the sample collection so that there's an association with what your health is like at the moment or has been recently with the samples at the time that they're given. And I know that it may be a little bit confusing calling it the GSD4 biobank, as you may know that, historically the foundation is and continues to support the APBD community. But we've recently tried to branch out to include families with other forms of GSD4, particularly the early onset. And so APBD is a form of GSD4, it's a late onset. So when we say GSD4, we mean all of the spectrum of APBD, all of the continuum of GSD4 related disease, and everything's associated with that GBE1 gene. So we're looking for samples both from our APBD patients as well as some of the newer families that have connected with us that have the other early onset forms of GSD4. And the purpose of collecting these biosamples is to use them in research. And so that can look like many different things, and I will get into that in my next slides. I also want to just point out that at this time, our biobank, which is supported through our collaboration with Combined Brain, which is a consortium of different patient advocacy groups that all are rare diseases in the neurological disease space, so neurodevelopmental disease as well as neurodegenerative disease, is only able to support US patients and participants at this time. But Combined Brain is looking to expand to other countries soon. And hopefully that will be something that we can reach out to our global community with sample collection in the future. So why is the GSD4 biobank important? So with collecting these biosamples, one of the challenges that many rare communities face, and ours is not excluded from that group, is that our patients are everywhere. They're geographically dispersed. They're in the United States. They're in other countries. We have a global population of patients. And with that dispersion, whenever people participate in clinical trials at a research institute or an academic site, those biosamples that are collected as part of those studies end up living in a silo with that institution, meaning that the researcher that collected them has them there. And while they can do more research or they can share them with other researchers to try to have a larger sample set of patients to include in a study, there's a lot of hurdles and challenges to sharing those samples across academic and research institutions. And so, what the foundation hopes to achieve through this is by having a foundation led biobank with a centralized biorepository to store these samples. It means that we can collect more samples from more patients, at least for now, across the United States and hopefully globally and internationally in the near future. And so, by having this larger set of data, why the biobank matters is that we have more information, more samples available in one spot. So it means less paperwork to find samples across different institutions. And this we really hope will facilitate a lot of research in APBD and GSD4. And so, what the biosamples can be used for and the type of research that it can support is research to help better understand disease, besides changes in GBE1, are there other changes in the body? Are there other systems other than glycogen metabolism that are impacted with APBD and GSD4? Those kinds of questions could be answered. It also, which is the main drive for all of us, is supporting the development of new treatments and the discovery of new treatments. And so those biosamples can be used to evaluate whether it looks like a new treatment might be beneficial to APBD patients by testing them in those patient samples. And ultimately, too, we hope that we can improve diagnosis and the staging of the progression of disease. And so, what that really hinges on is doing biomarker identification. And in this case, biomarkers could be, markers that help us identify when disease occurs so that we can get diagnosis sooner, so we can reduce the time of that diagnostic odyssey that so many of our patients are faced with, as well as to better understand and have something that could maybe better define what level of progression a patient has with their APBD. Is it earlier disease? Is that if we find a marker that is maybe less intense at that earlier stages, but then accumulates over time, and maybe we could get a better picture of the different stages and progression of APBD from those samples. So, reaching out to the community, everyone wants to know what are the potential benefits of participating in the biobank. And so, the most obvious answer is that it's a way to contribute to research in APBD and GSD4. So everything I had mentioned previously on the previous slide, all of that research can be supported through the biosamples that are being contributed to the biobank. And it's a really important resource on so many levels for when it comes to finding funding to support research projects, to get interest from pharmaceutical companies, to know that we have these resources available that can help support their research into understanding if a treatment will work. All of that is facilitated by your contribution to the biobank through your biosamples. This also can help with contributing to specific research studies. And by participating in the Combined Brain Consortium, the foundation has the opportunity to participate in studies that Combined Brain is working to support. And those are both, can be individual disease-based studies as well as cross-disease with similar diseases that are participants and represented by other patient advocacy groups within that larger consortium. And that could get at things like what I had mentioned previously about looking for biomarkers for disease, either within GSD4 and APBD, or maybe across some similar diseases, as well as the potential to do some drug repurposing studies with industry partners that the consortium has made. And those studies would be looking for drugs that have already been FDA approved and are understood to have some level of safety to see if they could be used to treat another disease like APBD. So having those biosamples available can make all of those types of research studies possible. Also, it's a way to support the community and the foundation. So in addition to fundraising efforts to support specific research projects or research studies, as well as the foundation, can support the foundation and the APBD community by providing those samples to the biobank. And the long-term benefits that we really hope to see come out of this initiative is those long-term research goals of identifying treatments, therapies, and cures, finding a way to diagnose earlier and faster so that we don't have this prolonged period of misdiagnosis that often occurs with APBD. And we can hopefully gain a deeper understanding of the disease. Like I said before, what other processes may be involved beyond what we know is disrupted with glycogen metabolism. So one of the main questions when it comes to providing samples or data, particularly in this day and age, is my privacy protected? And the straight answer is yes. So there's several ways that privacy is protected with participation in the biobank. The first of which is the clinical research ID is used to code the samples. So we've talked about the CRID, and we had a presentation previously from Combined Brain about setting up the CRID. Essentially, what that's doing is creating a unique ID that is used in place of your personal information to code the samples. So whenever your samples go into the biobank, if anyone requests samples out, they see your CRID number, not your name, not your birth date, none of that personal identifying information. And that CRID is set up by you as the participant, so you have the control over who has that information and who can connect it to your personal information. There's also just generally a lot of strict regulations of federal regulations regarding privacy protection in biorepositories and biobanks. And part of that is ethical oversight. So, if you've participated in clinical research studies before, you may have heard of an IRB or an institutional review board. So all of the protocols that are used for collecting samples, storing samples in the biobank, protecting the patient's privacy of their samples and their information is all governed within that IRB protocol that needs to be approved through that outside organization. And Combined Brain also has their own governance committee that reviews requests for the samples, requests for data, and generally reviews the protocols on how samples are collected and maintained within the biorepositories that are supported through the consortium. And any data that's collected, like health information or surveys, those are all stored on secure data systems. So there's limited access. Only appointees and specific individuals are allowed to access that information. And there's different levels, and there's a lot of restriction on who could see anything that would correlate to your personal information. So with that said, what does participation actually look like? So if you reach out and say that you're interested in providing your biosamples to the biobank, the first step is connecting you with Combined Brain so that they can consent you into their biobank collection protocol. And so the first step would be filling out consent forms and then connecting or creating the clinical research ID or CRID if you haven't created it already. And that's the first steps into protecting your privacy and before any other information or samples are collected. So the next step following that is filling out some of those health surveys so that we can capture how you're feeling, how you're doing at the moment and around the time when your samples are going into the biobank. And that helps to categorize the different samples that we may collect. So we know if someone's experiencing certain symptoms and someone else isn't, then that can kind of be navigated through with the samples. So we know a little bit more detail about what's going on for the individual that donated those particular samples. You have the ability, and you'll be requested to upload your genetic report. That data is curated, so whenever data is shared, your actual genetic report would not be shared. It would just give the information of the mutations that were identified on your genetic report and what the impression on the genetic report was. So if it's a lot of times those reports will say, indicative of APBD or GSD4 or whatever the language the particular company uses. You also would have the option, if you choose, to connect your electronic health records. So what that means is connecting to the systems like MyChart or what have you that your doctor's office is using to keep track of your medical information. And like I said, that's up to you, whatever you feel comfortable with. If you want to participate in the biobank, you can choose to connect that information or not. It can be really helpful if you have, especially if you have a extensive history through your providers, to connect that information, because it's just more data that can be connected to your sample to better classify, whether you have GSD4 or APBD, what symptoms that you've developed over time, when they've shown up, and all of that information, any medications you may be on. So once all that information is collected, the final step is scheduling a visit for a phlebotomist to come to your home and collect the samples. So a collection kit is shipped to your home. The phlebotomist is only going to collect the blood samples. If you provide a urine sample or collect the nasal swabs, you can do those yourself, either before the appointment or during the appointment. But the nice thing is that instead of having to worry about finding a way to ship those samples back, or connect with FedEx at the right time, when the phlebotomist comes to your house, they'll collect all the samples, the blood samples they collected, and any urine or nasal swabs, box them all up, and take them with them to send back for them to be stored in the biobank. So I next wanted to address some common concerns that come up when participating in this type of study and this type of initiative. So one of the main questions that people will ask is, who controls how my samples and data are used? And so as I mentioned previously, there's a lot of governance committees, there's a lot of ethical committees that are responsible for evaluating how the samples are collected, how they're shared. So for our biobank in particular, if any researcher or a pharmaceutical company that's looking to study APBD or GSD4 were to reach out to Combined Brain and say, hey, I'd like to collect samples from the APBD Research Foundation to study this aspect of APBD, or look at this treatment for APBD, or look for a biomarker for APBD or GSD4. That request comes to governance appointees that are specified for the foundation. So members of the foundation that have gone through all types of ethical approvals for managing patient data, they review those requests for any samples or data from the biobank. And the main thing is that the foundation representatives then determine, does the study align with the mission of the foundation? Will it benefit patients in our community? And is it a reasonable request? So are they asking for too much of a sample? Does it seem like what they're asking for is the right amount, or are they looking for to use the right type of sample? So all of that's evaluated before a request is approved or denied. In addition, for the data aspect of that, so the health data and the survey data, there is that Combined Brain Governance Committee, which includes a number of appointees from the patient advocacy groups, and they meet to discuss the requests and determine whether or not that data should be shared with the entity or the researcher that's requesting that information. So everything's reviewed. It's not just like walking into a warehouse and pulling samples or information off a shelf and going through the checkout line. There's a lot of controls in place to make sure that what's being donated, these precious samples, are used for the right purposes. The second question that people may ask is, will this affect my care? So participating in the biobank isn't going to impact your current care plans. That's not what the purpose of this biobank is. So with that, with the information being de-identified, if samples are shared with a researcher and they're doing a particular research study, they don't know who that sample belongs to. They just have that number, that clinical research ID that's connected to the sample. So that also means that any study that would come back that would maybe evaluate certain markers, it cannot be connected back to you. So you would not get that specific information back for you. It would be treated as the group as a whole. And lastly, a question that comes up a lot is, can I change my mind? And as always, participation is 100% voluntary. We'd love for everyone in our community to participate and provide samples, but we know that everyone has their own personal concerns, their own limitations on what would allow them to participate. So whether you choose to participate or not, that's 100% up to you. And even if you choose to participate and decide at a later time, I don't really want my data to be shared anymore, or I really don't want my samples to be in the biobank. That's a process that we can work through to remove the samples from the biobank. So you are not required to continue to participate after you've provided those samples. So I wanted to end on, what we're ultimately looking for is for your contributions to the biobank through these biosamples to have a lasting impact. And what that means is that by participating in the biobank today or next year, tomorrow, 10 days from now, is that we're hoping that all of this leads to new breakthroughs for APBD and GSD4 by advancing science, supporting the community, and leaving a lasting impact, which a lot of is going to be supported through research studies that can be conducted by having these biosamples. So I want to extend my thanks. I know a number of you have already completed the Biobank interest survey, and I'm very appreciative of everyone that's taken the time to fill that out. If you haven't already, or if you've been on the fence about whether or not you want to participate, or it hasn't really been clear what participating means, if you feel more comfortable with it now, I invite you to take that Biobank interest survey. I'll drop the link in the chat, as well as a link first to our article walking through how to set up the clinical research ID if you think these are things that are interesting and things that you would like to participate in. And again, as always, if you have any questions, please feel free to reach out to me. I'm always have my phone on me, my email's on my phone. Please reach out if you have any questions, and I'm happy to help you either complete the surveys, work through your CRID, or answer any questions that you have. And so with that, I'll stop here, and I'm happy to take any questions you might have.
There's a question in the chat room right now.
Could you read that out to us?
Yes, yes, it says, from when will it be possible to participate from Europe?
That's a really good question. And that's the question that we've been asking because we have quite a few patients that we've connected with and families that we've connected with in Europe. So I don't have a clear picture of when Europe will be on Combined Brain's radar for expanding that IRB. I believe there's some efforts to look at South and Central America because there's quite a few groups in those countries now, and some patient advocacy groups that are from those areas that currently are participating. And so I think it's going to be a stepwise and decision as going through. So, the main limitation there is that actual IRB approval for how the samples are collected from the biobank. And so how that's written for Combined Brain right now is collection within the US. There might be
Lindsay,
a possibility. Mhm.
I have a question, very directly related. I've had two people who are in Europe ask if they come to the United States, could they be scheduled to give samples while they're here in this country, even though they are actually citizens and residents overseas? Do we know the answer, or could you ask that of your
Yes,
people?
I can follow up to get some more clarity on that, but I know in the past that there have been conversations related to that. And so, yes, that is a possibility. So, particularly if you're having a longer visit, because coordinating the collections, or if we can coordinate before a visit, that would probably be the best. But the collection has to be in the United States. So they can't coordinate with someone to collect in Europe. But if you are visiting from another country or another area and are going to be in the United States, we could set up a collection then.
Okay, super. Any other questions in the chat that we need to address? Because I have a question that I'm hoping our people in the room could answer. Any more chat questions? Okay. For those of you who have not signed up to get let's start with the beginning part, the CRID, the identifier that removes your name from the process and substitutes a series of digits that are kept very closely locked down. For those people who have not gone through that process, can you give me reasons that make it difficult for you to do that? because we need to lift those reasons away. Anybody who has not and feels comfortable sharing, unmute and let me know.
I
Okay.
also wanted to add, if someone doesn't feel comfortable speaking, feel free to add a comment in the chat box and I'll read it out. I don't need to identify a person. I'll read the question or your comment.
Okay. Thank you. Good job, Vanessa. Yes. So again, if you have not gotten a CRID, one of those research identifying numbers, and you're willing to share, please let us know through the chat right now, or is it to you, Anessa? Is that how they're going to do it?
They can just put it for everybody if they want to put it for me, or they can do that. It doesn't matter. I'll read it out.
All righty. Second question to everybody in the room now. We're trying very hard to have a general level of understanding of how important this is. We have 5 patients now who have completed the biosampling process. They've given the biosample, they completed the little survey that went with it, the health survey, and we're trying for 15 more. I would love to see that before the middle of 2026. I really would. That would be ideal. If you're on the fence and you're not quite sure that you would do it, can you tell me what would impede you from doing it? Either tell me here on the screen or again through the chat, because we need to know that we're getting through to the group, and if there are concerns or there are impediments that we're just not thinking about, we need to address them. Because this is a big deal, guys. It sounds so simple and it sounds like such a pain in the neck, but it's huge for us. We're telling the world that we can organize, that we have samples, that researchers can do work on our behalf, that we are going to remove a lot of paperwork. The borrowing of biosamples from one university to the next is very complicated. Takes a lot of time. And if we're in the middle and we can just do it, it means that research can proceed so much more quickly. So this is your opportunity again to unmute yourself and say what you think is impeding you, or to add it into the chat area for Anessa to retrieve. Anybody want the floor? Okay.
This is Aura. I just wanted to echo that exactly. You know, in my professional life, this is an essential. These kinds of consortium are what actually move the dial, and we have established and continue to establish just greater and greater governances over privacy and use of data and use of information, such that I think that if those are the concerns that you have, that really can be addressed and be addressed in a way that you should be able to feel very comfortable about that. And then there is unquestionably power in numbers, and having been in clinical trials where we're trying to do just that, get one institution to share with another institution. It is very labor intensive, it is very time intensive to get these data use agreements that they're just kind of sharing between one and another. And so this has become the modern model. This is how we scale, and how we take our rare disease into something that is leverageable into a number that matters to pharma, that matters to funders, that matters to really be able to try to get changes to our disease state. So that's just my soapbox, and reiterating for everything that Lindsay said and that Harry's plea there.
Or I couldn't ask for a better spokesmodel there. Thank you. Thank you. Deborah, I'm going to put you on the spot, because I believe you have contributed to the biosampling and the biobank. Can you tell us what it was like.
Sure. It's easy. It takes a few minutes to fill out the forms. That's probably the most time consuming, but again, very simple, very straightforward questions. And then the rest of it is a breeze. They send you a box with everything you need to do. You give a urine sample, they come and do a blood test, you do a nasal swab, and that's it. It takes a few, maybe 10 minutes when the person comes to your house or apartment. Um, it's basically paperwork. It's nothing complicated. It's just about different aspects of you and your health. It's simple, and it provides a tremendous amount of information to ultimately help you, to help us find treatments, to find cures, to enter any research protocol. It's the basis of what we all need to do.
Deborah, thank you.
It's safe and it's easy.
Okay. And you've been with us, you've been a founding member of this chat group since, gosh, God was born, right?
Yes.
And this is a first for us.
This is a tremendous step forward for those of us who can no longer step. This is the way you can do it. No, it's a very small and necessary component of putting GSD4 and APBD on the radar, because once we have a storage of data, these are our people, this is what we found, this is what their biosamples look like. It's really helpful for any research going forward.
I want to clarify with Lindsay, there is no cost to the donor. Is that correct? That's solid.
Yes, that's correct.
So the cost is
From the
boogers
foundation. Yeah.
It's the monetary cost is borne by the foundation, and the patients are providing just the biospecimens themselves.
Right.
Okay.
Harriet,
Wonderful.
Harriet, we have a few responses here in the chat. If I could read the first comment, and then another one has a question. So a comment, I have no objections to participating. I was only diagnosed within the past month, and I'm just finding my way within the APBD community. I will probably sign up in the next few weeks. That's wonderful to hear. Really great. And then a question here is, when will we move ahead collecting samples from the next group who already expressed interest and done the consent?
Lindsay, that's yours.
So that is my question. Yes. So I'm going to be reaching out to the individuals who have already completed the Biobank interest survey later this week or next week. It's been on my list of to do, and there's been a lot of things added to that to-do list. So I wanted to make sure that all of the information was as clear as possible, and no information has changed, because there have been some minor changes to the processes with improvements. They're changes, but they're improvements with the new year. So I wanted to make sure I was covering all the bases before I started reaching out to get everyone engaged that has already reached out. So that will be happening very soon.
Mm-hmm. Here's a comment, perhaps some feedback. As one person who has contributed to this biobank, a few areas in the collection process could have been clarified. And then it says, here we have a few suggestions. One, should the participant fill out the paperwork in advance? Two, what samples can the participant collect in advance of the, oh gosh, I'm gonna butcher this,
Phlebotomist.
Okay, nasal swab. Okay, a urine sample that has been immediately labeled with the time and date of collection and refrigerated after collection, and for long can it be, and for how long can it be refrigerated? Does the nasal swab have to be refrigerated?
I can answer some of those questions. So they give you ample time frame. So they'll say, I'll be there between 11 and 12. My recommendation is you do your urine sample around that time. so that you can just do it, or shortly before they get there, or when they get there. They're with you for 10 minutes. So it would be very helpful to fill out the forms prior to them getting there. They're really there just to do a blood sample, collect the urine sample on the nose swab. So have everything done prior to them getting there. Ideally, do everything within an hour of them getting there, and then nothing has to be refrigerated. You could just give it to them. It's very simple.
Lindsay, why don't you add that into your list of things that go into the packet that you're preparing?
I've copied down that response. Thank you so much for pointing out some of these questions and issues that you ran into. I will make sure that, these are the things that we're looking for with the pilot group that went through those first 5, is to see where there's some hiccups and, where we can improve the process with how we're connecting. And I will be fully transparent. It wasn't just a learning experience for the patients, it was a learning experience for me as well. So, there's a lot of parts of this process that are much clearer to me now. And so I'm hoping that I can pass that on to clarify things as we move forward with collections for the biobank.
Yeah.
And Lindsay, there's another comment as well that's related to that. I
Okay.
can read it, or if you want it to, it's a very good comment.
Why don't you just read it?
Okay. Is a first morning urine sample better than one that is collected later in the day and may be diluted from fluids taken that day?
That's a really good question. I will follow up on that. I believe when I had asked about that before from Combined Brain, I don't think they specify, because one of the other means by which they collect samples is at family conferences for the different patient advocacy groups. So a lot of those times, those collections are throughout the day. And most people are probably not going to be wanting to walk around a hotel convention center
Right.
with samples in their purse or in their pockets or in their bags.
I'm
So
fairly
generally
certain.
they're
I'm
collecting
fairly
throughout the day.
certain they did not request or require a first sample of the day.
Mhm.
Again, great things. These are details that one never thinks about until you're at that moment and you say, oh my goodness. So thank you for getting them to clarify, and then it'll just put the whole problem to bed. Some other comments here, questions. Well, we're going to have you back again, Lindsay. No good deed goes unpunished. We need to have you complete this presentation numerous times, because we do have new people who join us. You're on record so that people can see through the website what you went through. But I just know that more is better, and a personal approach really does help to make the impact. So let's all wave goodbye to Lindsay. Thank you so much.
Thank you, Lindsay.
For being with us today. And the rest of the group is going to stay with me